Our next speaker is a physician scientist with over 10 years of international experience in clinical research and development, regulatory and medical affairs, and program management of vaccine discovery and development in Asia Pacific, Europe, Latin America, and Africa. He's a global medical director for Takeda's vaccine business unit, a TED educator, and a digital health expert at the World Health Organization. He has developed TED lessons on influenza, tuberculosis, meningitis, and coronavirus, which have been translated into 24 different languages and viewed by millions globally. As the first Gates Global Health Fellow from Asia at the Bill and Melinda Gates Foundation, he launched the Collaboration for Tuberculosis Vaccine Discovery, an international network of over 70 institutions from all over the world dedicated to fostering collaboration in TB vaccine discovery. Dr. Sanicas is a part of the Safety Platform for Emergency Vaccines project that creates resources and tools to facilitate COVID-19 vaccine safety, clinical trials, and pharmacovigilance. Dr. Sanicas earned his bachelor's degree, cum laude, and MD from the University of the Philippines, Manila. He has postgraduate degrees in vaccinology and clinical development, cum laude, Università di Siena, Italy. Infectious Diseases, London School of Hygiene and Tropical Medicine, UK. Health Economics, University of Aberdeen, UK. Clinical Risk Management, University of Leeds, UK. And Public Leadership, Harvard Kennedy School, US. Dr. Sanicas is a Fellow of the Royal Society of Tropical Medicine and Hygiene, and a Fellow of the Royal Society for Public Health, and a Fellow for the Royal Society of Arts. Let us now welcome our next speaker and my friend, Dr. Melvin Sanicas.
Good morning, good afternoon, everyone. First of all, thank you to the organizers for inviting me to be part of the EuroHacks 2.0 postgraduate course. Just over a year ago, before the pandemic started, vaccines were not really considered something worthwhile to talk about, and people were, for the most part, vaguely familiar with vaccines. But since the end of 2020, COVID vaccines have been in the spotlight and have become an everyday topic of conversation. And almost everyone now has some idea of the principles of vaccination, and that's good. But it also has made vaccines the topic of lots of misinformation and disinformation. And it's always a challenge to talk about a topic as popular as vaccines are at the moment. But I will try my best to squeeze the important bits in the next 30 or 40 minutes. First of all, some disclosures. I am employed by Takeda in the vaccines business unit, and I work as a technical consultant or an advisor for some of the groups and companies listed here, but I'm not representing any of them today, and I am here in my personal capacity as a physician and a scientist who has been working in the field of vaccines and vaccinology for over a decade now. So 1 year and 4 months since the pandemic was declared, we are now here. And I honestly thought that because we already had vaccines in December last year, we would be in a much better situation by now. But the vaccines have been distributed mostly in the rich countries. And although there are signs that the cases are decreasing, I don't want to be too excited because the cases have decreased 3 other times in the past. Yet we are here again in a situation where it seems like the outbreak is not yet ending anytime soon. But the other thing that we should be hopeful about is the fact that unlike this time last year, we now have drugs that have proven to be effective in reducing mortality or reducing hospitalization or reducing death or hospitalization from COVID. My talk today will have three parts. First, the different vaccine types or technologies, COVID vaccines that have been approved by at least one national regulatory authority or NRA, and SARS-CoV-2 variants. And I hope that these three topics, with these three topics, I will be able to cover as many subtopics in my talk, and hopefully you will have more specific questions after. So firstly, the different vaccine types or technologies. The two main traditional approaches are, as we know, the inactivated vaccines and the live attenuated vaccines. So when we talk about inactivated vaccines, these include most influenza vaccines and the inactivated polio vaccine. They contain a version of the organism that has been killed or inactivated using chemicals, heat, or radiation. And the killed form acts as an antigen and stimulates the body to create an antibody response. Inactivated vaccines can be given to people with compromised immune system. It is a well-established technology with defined pathways for regulatory approval. However, it requires special laboratory facilities to grow the virus safely before it can be inactivated or killed. Then we have the live attenuated vaccines. So this include measles, mumps, rubella vaccines. They contain attenuated or a weakened form of the disease-causing pathogen. They can still replicate, but they are not able to cause disease. The attenuated organism acts as an antigen and stimulates the body to create a robust antibody response, but it may risk causing disease in people who have weak or compromised immune systems. There are no approved live attenuated vaccines for COVID, but there are candidate vaccines being developed. It is a well-established technology as well, with defined pathways for regulatory approval, but like the inactivated vaccine, for live attenuated vaccines, you require special laboratory facilities to grow the virus first before you can weaken or attenuate them. Then we have the protein subunit vaccines. They do not contain the organism itself, but use fragments of protein from the pathogen as antigens to trigger an immune response, and doing so minimizes the risk of side effects. But also this means a weaker immune response as compared to vaccines using the whole pathogen. Because of this, subunit vaccines require adjuvants or substances to improve the immune response. An example of an existing subunit vaccine is the hepatitis B vaccine. And there are over 60 protein subunit vaccines against SARS-CoV-2 under development, including the RBD trimer of S protein, S1, recombinant S proteins, M proteins, and so on and so forth. This type of vaccine technology is also well established. It's relatively stable. It's relatively complex to manufacture, and you almost always need adjuvants and boosters to make sure that the immune response is durable. And it takes a while to really determine the best antigen to use for this type of vaccine. Then we have the viral vector vaccines. These vaccines use either a harmless virus or plasmids. And these harmless viruses are used to give genetic instructions to produce antigens. Genes of a pathogen are inserted into the genome of the vector, which infects host cells and then travels to the nucleus to express the genes of the pathogen. Viral vector vaccines can mimic natural infection and trigger a strong immune response. And vectored vaccines can be replicating, where viral vector is produced and are able to infect new cells, or non-replicating, where the vaccine antigen is produced, but the viral vector cannot be reproduced. And this is also a well-established technology. We have existing viral vector vaccines that have been approved in the market. The immune response is robust, and it involves B cells and T cells. However, previous exposure to the vector could reduce effectiveness, and it's also relatively complex to manufacture. Then last but not the least, we have the DNA or the RNA or nucleic acid vaccines that use genetic materials, either an RNA or a DNA, to provide cells with instructions to make the antigen. In the case of the mRNA vaccine, it contains the messenger RNA, a single-stranded RNA molecule that complements DNA and is created when the DNA is transcribed by RNA polymerases to create mRNA. Once the genetic material enters the human cells, it uses our cells as machinery to make the antigen that will trigger an immune response. Because the antigen is produced inside our cells and in large amounts, the immune response is expected to be strong, as we've seen in the mRNA vaccines available at the moment. The existing RNA vaccines need to be kept at ultra cold temperature, which may prove to be challenging for countries in low and middle income countries. It's a relatively easy to manufacture these kinds of vaccines, and booster shots may be required. So now we move to the COVID-19 vaccines that have been approved by at least one national regulatory authority. So this is just showing us that in June last year, there were 130 vaccine candidates and 17 of them were in clinical trials. And at the moment, there are over 100 vaccine candidates in the different stages of clinical development, phase 1, phase 2, phase 3. And I will be discussing here some of the vaccines that have been given approval by at least one national regulatory authority. So we start with the first vaccine to be approved, which is the CUMMERNATI or the Tozinamiran or BNT162b2, which is the mRNA vaccine developed by Pfizer, BioNTech, and Fosun Pharma. The efficacy is 95% against symptomatic disease, and it's given 2 doses 3 weeks apart intramuscularly. As we all know, this is the vaccine that needs ultra cold chain. However, studies have been done, especially in Europe and Israel, and now they are able to say that the vaccine can be stable at room temperature for at least 30 days. And because this is the vaccine that has been used in many developed countries, we have lots of data. And we have seen data comparing this to data using this in countries that have many of the variants, and the vaccine has proven to be still effective in places where the variants were circulating. And one good thing about this vaccine is that it is being used in some countries in the UK and in Spain in a sort of a mix and match type of study, where the first dose could be either Pfizer or AstraZeneca, and the second dose could be either Pfizer or AstraZeneca. And it seems like this is going well, and preliminary studies in Spain have shown that it is okay to mix and match these two vaccines. And we are waiting for the results from the Oxford COMCOV study. And then we have the Moderna vaccine, which is also an mRNA vaccine. The efficacy is vaccine efficacy against symptomatic disease is 94.1%, and it's given in 2 doses, 4 weeks apart, also intramuscularly. Also needs ultra cold chain, but not as cold as Pfizer. And the developers are also trying to optimize the vaccine so that, like the Pfizer vaccine, it can be used in normal temperature in at least for at least 30 days. And this is also a vaccine that's been used in many of the developed countries. So we are seeing a lot of studies, data from the studies showing that the vaccine is effective up to now. So it has been used in many countries for at least 8 months now, and it is still effective. So that is a good sign. In March, they started testing in babies and young children, and they also started a trial to see whether a new formulation specifically for the B1351 variant will be as effective as the original formulation. Then we have the AstraZeneca vaccine. It's called Vaxzevria, or the AZD1222, or Covishield in India. The efficacy is around 62 to 90%, depending on the dose. But the study done in US showed that the efficacy is actually around 80%, 79, 80%, and that is good. It's a 2-dose regimen given 4 weeks apart. Studies have shown that if you increase the interval from 4 to 7 weeks to 8 to 12 weeks, so in other words, if you give if you increase the interval, vaccine efficacy is better. It is stable in refrigerators for at least 6 months, and this is the vaccine that's been used the most globally, because this is the vaccine that's being distributed in the COVAX facility of WHO. So in South Africa, there was a small trial that failed to demonstrate protection against the variant there. So the South African authorities stopped using this vaccine for a while. And AstraZeneca in Oxford announced a few months back that they will start trials in children as young as 6 years old, and they are also working on a new version of the vaccine tailored to this B1351 variant. As you know, this is the vaccine that's been implicated in several news articles and news coverage in the media a few months back because of the association with blood clots, and we now know that the incidence of cerebral venous thrombosis, or CVT, in AstraZeneca vaccine is around 5 in 1 million doses given. And in mRNA vaccines, it's 4 in 1 million doses. And we know that the incidence of CVT in COVID infection is much higher, which is 39 in 1 million. Then we have the Sputnik V vaccine, also known as GAM-COVID-Vac. The efficacy is 91.6%. It's a 2-dose regimen as well, given 3 weeks apart. It's given intramuscularly. It's a combination of 2 adenoviruses called Ad5 and Ad26, or adenovirus 5, adenovirus 26. We have seen the results of their phase 3 trial, and it's pretty good. They are also doing a study trying to mix this with AstraZeneca vaccine to see whether the efficacy can be increased, and they are doing a single-dose version of this vaccine called Sputnik Light. And the study just wanted to see whether 1 dose of this vaccine is actually enough. Then we have the Convidecia, also known as Ad5-NCoV, from the Chinese biotech company CanSino Bio. The efficacy is 65.28%, but it's only 1 dose. So I am sure if you give 2 doses of this vaccine, you will have higher efficacy. And it remains to be seen whether this will remain 1 dose. Because if you want to optimize protection, you should give 2 doses for this. But I guess this is what the company wants, to promote this vaccine as a 1-dose only dosing regimen. But we will know more as this is being used in different countries. So this has been approved by the Chinese regulators in February this year, and in March this year they started using an inhaled version of this vaccine in clinical trials. Then we have the Sinopharm vaccine. This is, by the way, the first Chinese vaccine that's been given emergency use listing approval by the WHO. Phase 1 and 2 of Phase 1 and 2 trials have been published in the Lancet, and several countries have been using this already and have been doing their own local studies. UAE gave full approval, announcing that it had an efficacy rate of 86%, and a preprint came out a few weeks ago showing that the vaccine is modestly weaker but still effective against B1351 variant. And we will most probably be seeing more data from this company since it has been given the go signal by the WHO. Then we have CoronaVac from Sinovac. It's an inactivated COVID-19 vaccine. Of course, the main advantage of this is that it can be stored in a standard refrigerator at 2 to 8 degrees Celsius. A randomized, double-blind, placebo-controlled phase 1/2 study was conducted in China, and in those in China in 18 to 59 years of age, and also in people 60 and over, 60 and above. And the results, the trials found that immunogenicity in most patients and was good, and that the vaccine was generally safe and well tolerated. At the time of this recording, phase 3 data has not yet been published in a peer-reviewed journal, but we know that this vaccine has been used in Turkey, Indonesia, and Brazil, and they have reported vaccine efficacy in these different countries, varying from 50.4% to 83.5% in Turkey. And this is given in 2 doses, 2 weeks apart. And we know that recently Chile has reported a real-world study in the country, and it showed that the vaccine is 67% effective against symptoms. It reduced hospitalizations by 85% and intensive care admission by 89% and deaths by 80%. And that is actually pretty good as well. Then we have the vaccine, Covaxin, also known as BBV152. It's the inactivated vaccine by Bharat Biotech and the Indian Council of Medical Research in India. It is formulated with a toll-like receptor 7 or 8 agonist molecule adsorbed to alum, developed by the Hyderabad-based Indian biotech company, Bharat, in collaboration with the National Indian Council of Medical Research. Phase 1 trial results show that the vaccine was well tolerated and it enhanced immune responses. And it also had pretty good phase 2 results. And the company reported via press release that efficacy is 78% in its interim analysis of its phase 3 trial. And that was a little bit lower than the initial figures announced. That's 80%, but we will see what really is the case when they publish this in a peer-reviewed journal. And then we have the Epivac Corona, which is the second vaccine from Russia. It is a peptide-based vaccine, stable in refrigerator for up to 2 years. It's given in 2 doses, 3 weeks apart. It contains small portions of the proteins of SARS-CoV-2. The phase 3 trial began in November. And just like the first Russian vaccine, Sputnik V, EUA was given before completion of phase 3. And we are waiting for the results to be published anytime soon. And then we have the Johnson vaccine, also an adenoviral vector vaccine. It's called Ad26-CoV2S. The efficacy is 72% in the US, 64% in South Africa, and 61% in Latin America, and this is just 1 dose. So this vaccine is using the same technology that was used in the Ebola vaccine that has been approved in 2018. And this vaccine was given EUA by the US FDA in end of February this year. So in summary, we have several vaccines that have been approved now by at least one national regulatory authority, and some of them have been given full approval by specific countries. For example, the Pfizer-BioNTech vaccine has been given full approval by Switzerland and the United Arab Emirates, and Sinopharm vaccine has been given full approval by the Chinese regulatory authority. And the ones below are the other vaccines that have been given EUA, but we are expecting that these vaccines will also receive full approval as more data comes in and more data is shared with the regulatory authorities. So it's actually good for us that we have 10 different vaccines approved for COVID. And this is unusual because we have never had this situation before where we have 10 different vaccines for only one pathogen, and we are using 4 different vaccine modalities. But the not so good news is that none of these companies can manufacture 7.8 billion doses, and most of the vaccine need 2 doses. So of course, this means that we will not have enough doses for everyone this year. Then we move on to SARS-CoV-2 variants. We know that that viruses mutate over time, some more than others, and that's just how viruses are, and that's normal. For example, the flu viruses, they change often, which is why we recommend that you get a new flu vaccine every year. And for RNA viruses like SARS-CoV-2, we actually really don't know whether we would need a booster, and this is something that scientists are following up. But anyway, we will be discussing here why this is something that we need to monitor closely. We know that if there are a high number of cases, there's increased risk of mutations, and if we have a situation where there is an increased risk of mutations, there are higher chances of variants appearing. And we know that not all mutations lead to variants, because some of the mutations are what we call the nonsense or the missense mutations, which will actually not change any of the amino acids in the protein. But if these mutations happen in a specific portion of the virus, and if they accumulate, then we will see these variants that we are now hearing a lot in the news. We know that all strains are variants, but not all variants are strains. So if you look at the cartoon here, you see the parental strain, you have mutations, and then you will have variants. If you have all these changes in the RNA, you will see variants, but it does not mean that it's already a strain, because for it to become a new strain, you need to see evidence of a change in the behavior of the virus. Is the virus transmitting more? Is the virus becoming more deadly? Is the virus able to evade antibodies produced by the vaccines, and so on and so forth? So we have different variants, and there are different possible answers to questions why these variants are emerging. First of all, it could be the inherent rate of mutation of the virus. It could be it might be happening because of fitness advantage. It can happen because of the presence or lack of immune pressure. It can happen because of intermediate hosts. So we know that coronaviruses can infect other animals as well, just like influenza, and that's one of the reasons why can see these variants emerging. And then again, as I've mentioned earlier, if you have more infections in a community, if the outbreak isn't controlled, then there's higher chances for us to see variants. So there are different variants, and these are classified into variants of interest or variants of concern or variants of high consequence. So if you have a variant that has been associated with changes to receptor binding, reduced neutralization by antibodies, or reduced efficacy of treatments, then it's considered a variant of interest. So basically, what we should remember here is that any variant that is associated with changes that are important are considered variants of interest. And we have lots of variants of interest. As you can see, the P3 variant, first discovered in the Philippines, is considered a variant of interest. Then we have variants of concern. These are the variants that have shown evidence of increased transmissibility, evidence of more severe disease, evidence of a significant reduction in neutralization by antibodies. And at the moment, we have 4 variants of concern. We have the B117 that was first discovered in Kent in the United Kingdom, B1351, which was first discovered in South Africa, B1, which was first discovered in Japan from people in Brazil who traveled to Japan. And then we have just a few weeks ago, the B1617 or B16172, the variants that have been discovered in India. And I was giving this same talk in January this year, and during the time there were only 2 variants. So it just tells us that the more this pandemic, the more that the outbreak is not controlled, the more variants we will be seeing. Then we have the variants of high consequence. So these are the variants that has clear evidence that prevention measures or medical countermeasures like masks or vaccines or hand washing have significantly reduced effectiveness. And at the moment, there are no SARS-CoV-2 variants that rise to the level of high consequence. So that is a good thing for us. I will be just showing in the next few slides results of clinical trials, just showing us that if you look at these vaccines, so here you will see the J&J vaccine. This was studied in the US, Brazil, and South Africa, and during that time, the variant in South Africa was circulating. And if you compare the vaccine efficacy between US, Brazil, and South Africa, you will see that the vaccine efficacy in South Africa is a little lower, but it's still working. So the key message here is that the vaccine is still working. And then it's the same thing for the Novavax vaccine that was also done in the UK. The clinical trials were done in the UK and South Africa. You will see the same thing, that the efficacy in the UK is around 86%, and in South Africa it's around 60%. Of course, it shows that the variant that was discovered in South Africa seems to be able to evade neutralization by antibodies produced by the vaccine. But again, we have to look at this in in another perspective, that the vaccines are still working. If the numbers are 10, 20%, then we should be scared. We should be concerned. But that's not the case here. So this is a figure that I created a couple of weeks ago. This is just showing us all the different variants where they were first identified and the vaccines that have been tested so far, either in a laboratory study looking at the neutralization of the virus using antibodies produced by the vaccine, or in clinical trials. And you will see here that this is a very good picture, a positive picture, because all these vaccines have shown that they are still working very well against these variants. And most of these are from real world evidence. So these are studies done after phase 3 clinical trials. And these studies were done in different countries, in Israel, Qatar, United Kingdom, Brazil, Chile, United States. And then, so what is the best case scenario is that all these different vaccines that I mentioned are still working and will still work in the future. And this is showing us results in several countries, in the UK, US, and Chile, and this was in April. And you will see that even in these countries where the different variants have been circulating, you will still see a very obvious effect of the vaccine, and the vaccines are still working. So that's the bottom line message here. And worst case scenario, let's say that really that there is a new variant that's totally able to evade the antibodies produced by the vaccines. What will happen now? So the good thing is that the vaccines can be modified to match the variants if required. And this will not need the phase 3 studies anymore. So for all these vaccines that have been approved, all those 10 vaccines that I have mentioned, you don't need to do the whole phase 3 clinical trials again. You just need to do immunogenicity studies, and then they can be approved. So it's similar to what's happening with the flu vaccines. So every year when we change the strains that are included in the flu vaccines, we don't do phase 3 clinical trials. We don't do phase 3 efficacy trials for the vaccines. We just do immunogenicity studies. And so I think here, this is one of my last few slides. I just want to emphasize that, yes, the virus is changing, but the path to ending the pandemic is the same, which is get vaccinated as soon as you can, vaccinate as many people as possible. And although this is like a race, right, between vaccines and the variants, we know that we are going to win in the future, just like all the past pandemics. It's just, I think, a change of the mindset, because people are always so concerned about variants, but they're not pushing for vaccination as much as they should be. And there is a mismatch there of our concern for these variants and the efforts that we are doing to increase or make these vaccines reach people as soon as possible. And I would like to end my talk by just emphasizing that, these vaccines are like the moon landing of our generation. They're like the most significant scientific achievement of our lifetimes. And I think we have not absorbed the enormity of the importance of vaccines. And I'm saying that because when you speak to people, when you look at social media, you see that most people's comments are still looking at the weaknesses of the vaccines, or basically not appreciating the vaccines for what they are. They are our solutions to end the pandemic. And I hope that you will be more active in your social circles, in your families, in your friends, and explaining to them how valuable these vaccines are to us in our situation in this pandemic. And I hope that you will be able to convince more of your friends and families to get vaccinated as soon as they are able to get the vaccines. So thank you very much, and I will be very happy to answer your questions.